and welcome to Night School. My name's Aria.
And I'm Darian.
And if you're new here, we're so happy to have you. Regulars, y'all already know this, but we are a program of nightlife at the California Academy of Sciences, which is an in-person event that mixes science, culture, and art every single Thursday night. We're the online version of that.
Much like Night School, coming back from hiatus in 2023, the subject of tonight's Night School is species that have come back from the dead, or species that were declared by Western science to be extinct before being rediscovered very much alive. They're sometimes called Lazarus species. Tonight, you'll meet three people who have either been the ones to rediscover a Lazarus species or who work on the conservation and regeneration of these species. It's not often we get second chances like this, but when we do, it can come with some fascinating stories and some important lessons. And so I hope you're all as excited as I am to hear from our guests tonight.
And I will go ahead and introduce them. We first will have evolutionary biologist, Dr. Stephen Gaughran, who joins us from New Jersey, where he's a National Science Foundation postdoctoral research fellow at Princeton University. And he'll be sharing the story of one single tortoise, specifically a Fernandina Island giant tortoise, a lot of long titles in here, that he discovered living after the species had been presumed extinct for nearly 100 years. Thanks to museum collections, Stephen was able to prove that this species story may not be over. And an additional important note, this particular tortoise individual's name was Fernanda, which is amazing. Nickname. And following, we have marine biologist and research associate at UC Santa Barbara, Dr. Jeff Goddard, who joins us from Maine tonight. But he first spotted the teeny tiny clam that he'll be sharing about on the California coast. Before Jeff and his collaborators' species description work, this clam was only known from the fossil record, specifically from Pleistocene deposits such as the Brea Tar Pits in LA. And now this clam is known to be a living member of Santa Barbara's intertidal ecosystem, which is so cool. And to close us out, we have Dr. Rachel Paltridge, who is a threatened species ecologist with the Indigenous Desert Alliance, who's joining us from the Northern Territory of Australia, which also happens to be the home continent of the elusive and adorably loaf-like night parrot. It's a bird. And this mysterious bird was surprisingly rediscovered after it was thought lost to science for over 100 years. Rachel will share that very story and about the many actions taken since by an incredible network of Indigenous rangers and scientists to identify locations of new night parrot sites and to protect the species moving forward. So really, really cool species that we're diving in here tonight.
Exciting stuff. As always, tonight's program is live, so whether you're watching on YouTube or Facebook or newly Twitch, say hi. Let us know where in the world you're watching from. If it's your first time joining us or you're a Night School regular. Regulars, just hello, we love you. We will have a Q&A after each speaker, so be sure to type your questions in the chat and we'll get to them after each presentation. And with that, we will pass it to Stephen Gaughran.
Sorry about that. Hi, everyone. I'm Stephen Gaughran, and I'm an evolutionary geneticist at Princeton University. Tonight, I'm really excited to kick things off with a brief talk about the rediscovery of a Galapagos giant tortoise species that was thought to be extinct. I want to start, though, by thanking the California Academy of Sciences for inviting me to give this talk and thanking you for coming. I also want to make clear from the start that what I'm presenting here was a group effort in which I played a relatively modest role. The other key players are the field biologists and park rangers in the Galapagos Islands at the Galapagos Conservancy, who have put years of effort into this and made the actual discovery of the tortoise. Also, Evelyn Jensen, who's a professor at Newcastle University and a world expert on Galapagos giant tortoise genetics. And Gisela Cocconi, my PhD advisor at Yale University, who has spent the last several decades disentangling the genetic and evolutionary history of tortoises in the Galapagos. So the story I'll be telling you tonight centers around the Galapagos Islands, a volcanic archipelago about 1,000 kilometers off the coast of Ecuador. It's famous in my field, at least, for being the place where Charles Darwin collected important observations that eventually led him to his theory of evolution by natural selection. The Galapagos are home to thousands of unique species, including iguanas, birds, marine mammals, and of course, giant tortoises. We think that the ancestors of all Galapagos giant tortoises floated to the islands from continental South America more than 1 million years ago, and the descendants of these tortoises gradually colonized the new volcanic islands as they sprang up. Over thousands of generations, the populations of tortoises isolated on each island grew more and more unique through the slow process of speciation. The new species started to show differences in the shell shape, behavior, diet, and other traits. As you can see in these two pictures, the shell shape can vary so dramatically that when Charles Darwin visited the islands in the 19th century, he wrote about how the locals could tell which island a tortoise came from just by looking at it. Starting in the age of European colonization, though, explorers, sailors, pirates, and whalers would stop in the Galapagos and pick up tortoises as food for their remaining journeys. Unfortunately, this led to a decimation of tortoise populations across the Galapagos. You can see in this picture here how many empty shells were left behind after a harvest, and that's just gives you a fragment of an idea of the sort of destruction that was going on in the Galapagos populations at the time. And then later, the introduction of rats, pigs, and other domestic animals further destroyed tortoise habitat, destroyed their nests, and even predated on the young tortoises. So this means that today, 3 species of Galapagos tortoises are vulnerable to extinction, 3 of them are endangered, and 5 species are critically endangered. We also know that at least 3 species of Galapagos giant tortoises have gone extinct. The Pinta Island tortoise, whose last surviving member was rather famous, Lonesome George, died in 2012, the Floriana Island tortoise, and the Santa Fe Island tortoise. And up until just a few years ago, there was a 4th species on that list of extinct tortoises. So Fernandina Island is the youngest island in the Galapagos. The island has an active shield volcano, making much of the terrain difficult to traverse and inhospitable. You can see in this satellite image how much of the island is barren, with only just a small sliver of green plants that are found around the volcano. Despite these difficult conditions, an expedition from the appropriately California Academy of Sciences went to Fernandina Island in 1906 and found and collected a Galapagos giant tortoise there. And they thought this suggested that the island was home to a unique species of tortoise, just like the other islands. They gave the tortoise the scientific name Fantasticus because of the highly flared edge that you can see here with the red arrow at the top of its shell. Then the scientists sailed back to San Francisco, where they prepared the specimen and stored it in the museum's collections. Unfortunately, after that expedition, no more tortoises were seen on Fernandina Island. This led pretty much everyone to believe that the Fernandina Island tortoise was now extinct. As I mentioned, though, the island is uninhabited and particularly difficult to explore. So a few biologists held out hope that some Fernandina tortoises survived on the island. Bolstering those hopes were occasional sightings of scat and maybe tracks that they thought could have been from tortoises. Then, amazingly, in 2019, a female tortoise was found on Fernandina Island. She was nicknamed Fernanda and transported to a tortoise rehabilitation center for protection. Although it was exciting to find a living tortoise on Fernandina Island, there was still a big open question of if Fernanda was actually a native Fernandina Island tortoise. And that's because tortoises in the Galapagos can float between islands or sometimes even be moved by humans. So it was possible that Fernanda came from a nearby island in the archipelago and not natively from Fernandina Island. She also looks somewhat different from that Cal Academy specimen, which made us cautious at first about calling Fernanda a Fernandina tortoise. To settle this question, we turned to genetics. My advisor, Gisela, had shown through decades of work and dozens of scientific publications that Galapagos giant tortoises from different islands are genetically distinct species. So from this background work, we knew that if we could sequence the DNA of Fernanda and compare it to the DNA of that Cal Academy specimen from Fernandina Island, and also to genetic samples from tortoises from other islands, we could figure out which species Fernanda actually belonged to. So we set out to sequence the whole genome, more than 2 billion base pairs of DNA, from Fernanda and 38 other tortoises. We took a small tissue sample from each individual, extracted their DNA, and sequenced their genomes, something not technologically possible just a few years ago. We then ran a simple test to count up how many differences there were between the genome of Fernanda and those of other tortoises. We can visualize the number of differences as genetic distance in this graph. So the lower the number, that means the fewer genetic differences, which means the two individuals being compared are more closely related genetically. Here you can see this little pink dot representing the genetic distance between Fernanda's DNA and the DNA of the museum specimen of the Fernandina tortoise. And then in purple, you can see the distance between Fernanda's DNA and each of the other tortoises that are from different islands. So with that low genetic distance between Fernanda and the Fernandina Island museum specimen, it certainly looked like these two tortoises came from the same species and that Fernanda really was from Fernandina Island. But just to be sure about this, we also built a phylogenetic tree of all of the tortoises. Now, a phylogenetic tree is something like a family tree, which shows the relationships between related individuals. And just as in a family tree, you can see how a person is more closely related to their sibling than to their second cousin, a phylogenetic tree can tell you how closely related individuals are based on their DNA. In our tree, you can see Fernanda there at the top, pointed out with the red arrow, and the 1906 Cal Academy specimen right underneath. And if you read this like a family tree, you can trace the line back from Fernanda to see the closest line that she connects to, the Fernandina Island museum specimen, which is her closest relative on this tree. So this conclusively showed us that Fernanda really is a native Fernandina Island tortoise. And I have to admit that up to this point, I thought there was a good chance that Fernanda was not from Fernandina Island and had just floated there from some other island. So I was pretty surprised by these results. But all of the genetic tests we ran consistently showed that Fernanda and the museum specimen were genetically related, and I finally became convinced that the biologists in the Galapagos had found a living Fernandina Island tortoise. Today, Fernanda is being cared for at the Tortoise Center on Santa Cruz Island in the Galapagos. Although she's estimated to be about 50 years old, this is hardly middle-aged for a giant tortoise who can live for more than 100 years. Since discovering Fernanda in 2019, our colleague at the Galapagos Conservancy, Washington Tapia, has led scientists and park rangers on 3 more expeditions to Fernandina Island to try to find more native Fernandina Island tortoises, with the hope of starting a breeding program for this species. So far, they haven't found another tortoise on Fernandina. But this doesn't mean that they've come away empty-handed. They've reported tortoise paths, tracks, scat, and even beds where the tortoises sleep, all of which seem to be from very recent tortoise activity. They have another expedition planned to thoroughly search the habitable area of the island for remaining tortoises, and we're all hopeful that this time they'll be successful in finding a companion for Fernanda. Although now officially not extinct, Fernanda is still the last known member of her species, leaving the Fernandina Island tortoise on the brink of extinction. Several other Galapagos giant tortoise species are also critically endangered and need continued conservation work to help them survive and hopefully rebound. Fortunately, our collaborators at the Galapagos Conservancy are on the ground carrying out several projects to help these species. And we're now starting to see several ways how genetic studies can serve an important role in these conservation efforts. For example, the Floriana tortoise went extinct on Floriana Island in the 1800s. By doing genetic surveys of hundreds of tortoises, however, Gisela and Evelyn have identified individuals that are hybrids between Floriana species and a species of tortoise from Isabella Island. 20 of these hybrids have been brought to the breeding center on Santa Cruz, where a program is now underway to breed these tortoises for higher Floriana ancestry that will give rise to individuals to someday be re-released back onto Floriana Island to bring tortoises there once again. So as the genomes of more tortoises from across the archipelago are studied, then we may even be able to discover more hybrids that are carrying genetic ancestry from other Galapagos giant tortoise species that have gone extinct. And from there, design breeding programs that would allow the reintroduction and, if you think about it, de-extinction of these species. And then in museum collections around the world, we're still working to sequence DNA from all sorts of specimens, with the hope of better understanding what diversity has been lost and what biodiversity could still be out there. So I hope I've shown you today not just that the rediscovery of Lazarus species is exciting, but that field biology, museum collections, and genetic studies all serve important roles in species discovery and conservation. As field biologists continue to discover new animals in the wild and geneticists continue to refine our methods, it will become even more important and useful to combine approaches for the sake of biodiversity conservation. So thank you all for listening, and I'm happy to switch over now to take some of your questions.
Excellent. Thank you so much, Stephen. That was really insightful. And I'm glad the California Academy had the collections to help you conduct this research. We have a lot of excellent questions from our audience, but feel free to continue asking them in the comments and we'll get to as many as we can. So I know you said that you had thought that maybe Fernanda might have floated to Fernandina Island before you saw like the more conclusive genetic evidence, but how common do tortoises travel between the islands? James wants to know if the tortoises can swim from one island to another. Is that a frequent occurrence?
Yeah, so the tortoises don't swim, or at least don't swim well, and they don't seem to, I would say, willingly enter into the water. But they can float if they accidentally get washed into the water somehow. So it is possible. We don't think that it's all that common, and that's one of the reasons that these islands have been able to have their own species evolve independently, because once tortoises get to a new island, they tend to stay there and not have any new tortoises come. So it's rare, but it is possible. And when I was weighing it against thinking about a species that we thought was extinct but somehow had reappeared, that seemed like the less likely explanation, but turned out to be the right one.
Exciting. And then kind of on that note, Lydia asks, do they float on debris or are the tortoises buoyant themselves?
So probably both can happen. Lots of animals can float on debris, and we think that's how they get to islands sometimes. But tortoises themselves can actually stay afloat in the water without swimming. And that's probably how they got from the continent of South America 1,000 kilometers to the Galapagos in the first place.
Mm-hmm. Do we know how old Fernanda is?
So she's estimated to be about 50 years old. So not a young tortoise, but tortoises do live for over 100 years. So still hopefully has many decades left.
Shelko wants to know, how do you find like a single Galapagos tortoise on an entire island? Do you use methods like trained dogs? Like how did that discovery take place?
Yeah, so a lot of hard work, and that's why they had looked on Fernandina before for the tortoise. They put a lot of effort into trying to track down the most likely part of the island that they might find tortoises in. And then it was really just a lot of hours of searching and trying to look for tracks and looking for any traces of the tortoise and finding the most likely areas that one could be in. And so that's what they're doing now. And it's why, even though they've had 3 expeditions since they found Fernanda and haven't found a tortoise yet, I still remain hopeful that it's just very hard to find them and that they'll still be able to find a couple who are living out there on Fernandina Island.
I hope so too, for all of our sakes. Michael wants to know if there are any resources you know of that people in our audience can go to either support conservation efforts or to learn more about the recovery and discovery of these animals.
Yeah, I would suggest going to the Galapagos Conservancy website. So there are collaborators down in the Galapagos who are really doing this incredible Galapagos tortoise conservation work. They run the breeding centers. They're the ones who are out there searching for the tortoises, carrying out the important various strategies for conservation. And they list all of those strategies on their website and also various ways that you can help out their nonprofit.
Great. I'm sure some of those links will end up in our comments pretty soon. Lastly, our final question. Lisa wants to know, what are the advantages of breeding tortoises under human care versus leaving them on the island to breed?
Yeah, so There are a lot of threats to very young tortoises, to both the eggs and to the hatchlings. So as I mentioned, there are some domestic animals that are now on the islands, and those range from rats that aren't really domestic but introduced to some livestock animals. And all of those can accidentally trample the baby tortoises or the eggs or even eat them. So there's a big danger to the early stages in the tortoise's life. But once they get to a few years old, then they're really in a safe state to be re-released onto the islands, and that's when they tend to do it.
Okay, excellent. Thank you so much for sharing with us. Up next, we will hear from Dr. Jeff Goddard.
Okay, can you hear me okay? Thanks for inviting and joining me on Night School. My name is Jeff Goddard. I'm an invertebrate zoologist, marine ecologist, and I'm going to talk about a chance discovery I made a few years ago in tide pools near Santa Barbara. I've been studying nudibranch sea slugs on the West Coast for most of my life, and one of my long-term study sites has been the boulder field here at Naples Point on the Santa Barbara Channel. A few times a year, I've searched tide pools and underneath rocks here for nudibranchs and their prey. Turning a rock here, we might see this ribbon-like soft coral, and what would catch my eye are these little orange blobs here next to the soft coral. Enlarge them, well, they still look like orange blobs, but if you put them into water and get a closer view, voila, there's a nice little tritonid nudibranch that specializes on the polyps of the soft coral. These are some of the species my sons and I have found at Naples Point over the year, along with some of the population fluctuations we've documented. For now, just notice in the upper chart the decline in a couple of northern species at the same time that southern species at the bottom increase in abundance. I'll return to these warm water years later in my talk. But returning to getting back to our rock, there's a whole world of organisms under these rocks. So we have some sponges here, a little snail here, some ascidians or sea squirts here, these crusty worm, polychaete worm tubes with little white coiled worm tubes growing on them. And here we have the remains of a bivalve, an oyster. Now, some of the other bivalves that you'll see at Naples Point include rib mussels, a little nestling clam showing us its red siphons here, another species of nestling clam, little neck or steamer clams, juvenile rock scallops, and on tops of the rocks, you'll see California mussels. In November 2018, I was out looking for nudibranchs with Robin Agarwal, turned a rock and saw a species of clam I had never seen before. The shell was only about 10 millimeters long, but with that long translucent foot, it definitely stood out. Robin got a nice little video clip that we'll watch here showing the clam's surprising, almost snail-like movements. Uh-oh. Okay. The clam was one of a pair, and I dropped what I was doing to get photographs, close-up photographs showing the tentaculate mantle covering most of the shell here, the off-center umbo or beak of the shell, and the wavy ventral margin, and of course that exquisite white knight blade of a foot. Being a pair and seemingly rare, I chose not to collect the specimens. I figured the experts on bivalves would recognize the species as new or not from my photographs. But before I did that, I wanted to check the taxonomic family of these. So the mantle tissue covering most of the shell, the outer part of the shell, is unusual in bivalves. And you can see it in the little nestling clams here on the left. And they're members of the family Galeomatidae. So I figured that was the family of my new find. And the Galeomatids are a diverse taxon, and similar to sea slugs, they have reduced shells, tentaculate and colorful mantles, and many are commensal with other organisms. So you can see across the bottom row of images there, there's a little clam with a peanut worm, a little clam with a sea cucumber, a little clam with a heart urchin, a little clam next to a sea anemone. And finally, there's a species of Galeomatiid clam that lives attached to ghost shrimps that live in the mudflats and the bays on the West Coast. But my clam wasn't in any of the references I had, nor was it on iNaturalist or anywhere else I could find. So it was time to contact the experts. This is Paul Valentich-Scott, a malacologist at the Santa Barbara Museum of Natural History and an authority on the bivalves of the West Coast of the Americas. His 3 monographs here total nearly 3,000 pages and cover about 1,600 species, and that's just bivalves. Paul had some suggestions for the identity of my clam, including Cymatio electalis here, known from Santa Barbara south. But to my eye, the shell shape just didn't match. That umbo is more centered on electalis than it is on mine. Another possibility he threw out was this one. Shell's a little more similar, but not quite the same, and it lacks the wavy-edged ventral margin that mine had. Paul threw out a couple of other possibilities, but none of them appeared to match. So we were both tending towards a new species, which is always exciting, especially when it's a cutie like this. But a sentence in the text for Cymatio electalis, his first suggestion, caught my eye. Cymatio cookie, from the Southern California Pleistocene is more elongate than C. electalis. And yeah, mine is more elongate with that off-center umbo. The posterior end of the shell is more elongate. So this caught my attention, looked into it. I dug up the 1937 paper by George Willett, turned to the description and illustrations of Cookie, and there was my clam. The shell shape just looked to me like identical. So I told Paul about that I thought it was the same, and he said he'd need a specimen in hand to be sure. So I hadn't collected either of the first 2, but I said, sure, I'll go back. I went back 2 days after talking to him, and I knew I could go back and find the same rock I'd found those 2 under. Well, no such luck. I really looked. 2 months and 9 trips later, I turned a rock, and underneath this conspicuous chitin, there was a clam that caught my eye, and it turned out to be a third specimen of the same species. So now Paul could have a specimen in hand. Yeah, success. I was pretty excited about this. Took a few close-up pictures of the clam, this time relaxed in a fluid. And Paul requested the original fossil specimens from the LA County Museum of Natural History. A few months later, I was back at Naples Point, turned a rock, and found another specimen, an empty shell that was only 8 millimeters long. That was finding a needle in a haystack, literally. So by this time, Paul had the type specimens of Cymatioa cookie from the museum. And he carefully compared the specimens I'd given him with the type specimens of the fossil, including all the fine details of the beak and the hinge area and the associated teeth, and determined that they were indeed the same species. So Cymatioa cookie, known only from Pleistocene deposits in Los Angeles County, making it a living fossil, or as we've heard tonight, a Lazarus taxon. And there are some famous living fossils. The coelacanth, long known from the fossil record prior to its the discovery, at least by Western science, of living specimens in the 1930s. It was thought to have gone extinct when the dinosaurs went extinct. Another famous living fossil are the dawn redwoods, which were once widespread in the Northern Hemisphere, a long fossil record ending in the Miocene, so at least 5 million years ago. They're showing the distribution before they disappeared. And then in the 1940s, a small grove of living metasequoia were discovered in one valley in China. Now, at the time, these examples were big news. So here we have a story of a tree whose family lived with dinosaurs. Our clam wasn't as old a fossil, nor as famous, but when we published our paper about it, it did generate a little bit of celebrity attention. So my buddy here, Leonardo, and maybe Cymatioa cookie will get a star on the Bivalve Walk of Fame. Okay, so how the small size and cryptic habitat notwithstanding, how has this clam eluded detection for so long in one of the best studied molluscan faunas in the world? So where's it been hiding all these years? Why hasn't anybody seen this before? There's been so much shell collecting in Southern California, including people interested in micromollusk. So this is where we return to those warm years. We return to Naples Point and those warm ocean years. So during this period, we saw the sudden appearance of species of sea slugs known from farther south, as well as some other types of invertebrates. And they arrived as larvae, settled out, and grew quickly to adults. Turns out that most bivalves also have planktonic larvae. So my hypothesis is that Cymatioa cookie during the same period also arrived with warm waters from the south, settled, grew more slowly than the slugs, and grew up, and then we detected it in 2018. So the fact that no living specimens or recent shells of it have been found until now suggests to me that the clam probably originated from remote, less collected stretches of the coast. And so my vote would be, the credit for that lovely photo of the bivalve larvae, but my vote for where the clam maybe is living and has been living would be on the Pacific coast of Baja California, where there are immense boulder fields that stretch literally for miles. So is it living under boulders down there, or farther to the south on mainland Mexico? So needless to say, Paul and I will be excited to hear about additional finds of the clam and to learn more about its ecological relationships. So with that, I'll say thanks for listening, and I'll be happy to take some questions.
Hi, Jeff. That was amazing. I'm officially obsessed with this clam, and I want to be as famous as possible. So anybody out there watching, spread the clam love. This is so cool. And we have lots of great questions, so I'm just going to get right into them.
Okay, yeah.
So first, we have a question from Mitzi. These specimens are so tiny. Do you use special equipment to see them?
For me to find them, no, just my eyeglasses. But Paul Scott would have examined the details of the shell with a microscope. Mm-hmm. No, if someone wanted to really search for this clam and ultimately find who it's maybe living with, in a relationship with, you'd probably turn some rocks and do some excavating. You might sift the sediments under the rocks. So without knowing who it's living with right now, it's hard to say, but you may have to do some sifting to find additional specimens.
Yeah, totally. So itty bitty. Yeah, and then Queen McMoo asks, if these species can survive this long, how long can the individual live?
Oh, that's a good question. Some of the clams are notorious. The larger clams, like the gooey ducks up in Puget Sound, and some other clams, are known to live over a century as individuals. Remarkable lifespans. This clam, I don't know. I'm guessing a couple years. And I said grew more slowly than the nudibranchs because it's got to make more of a shell. It's just a guess, though. So I would guess a few years, but for something like this, but that's unknown as well. So
yeah, lots of open questions to investigate, sounds like.
Yeah. It would have been nice, with it being so rare, it would have been I didn't do this, but it would have been nice to just hold some alive and study them in the lab, like what kind of offspring, do they actually produce larvae or do they make little juvenile clams? Things like that.
Yeah. And speaking of like unique behavior, that snail-like movement that the video showed, is that something unique to this clam family, or like is that something that we see with a lot of different clams?
Some of the clams can use their foot to move rapidly. Cockles will be, their shell will be closed up. And if you put a predatory sea star, you can touch just the tube feet to the flesh just barely showing on a cockle, and this big foot comes out and it literally pole vaults off of the bottom.
What?
Okay, but as far as I know, only the galeomatids actually move more like snails. These are clams that to me look like they want to be slugs or snails. And it was in the last part of the video that Robin got. The way its siphon was extended with two tentacles, it looked like a snail, but obviously has a different foot. But anyway, yeah, it's that emotion like that is mainly in that family.
Gotcha. Okay, that's really, really cool. And
yeah,
we have several questions wondering about their shell and their translucent color. So couple things here, like what makes them so translucent and so lightly colored, and are they like actually very delicate? Like, are their shells very delicate because they look so translucent to the eye?
The shell of the living specimens seemed fairly like porcelain, fairly stout to me.
Oh, wow.
The empty shell I found, been sitting there in the sediments for a while, it was a little more fragile and not as translucent. So it was probably starting to absorb some minerals from the anoxic sediments. The fossil ones looked remarkably recent, but kind of opaque. So the shells are fairly strong. But yeah, they're almost glass-like and they're translucent, at least when it's alive. And that would just be a difference in the way the crystal structure of the calcium carbonate. I don't know why it would be that translucent.
Yeah.
And then the flesh, what's up with that translucent foot with the white glands? We did have to find out. So when I first saw it, it was kind of nestled against the burrow of some organism. Its siphon was out over the burrow, but I don't know who made the burrow. Is that clam always in the shadows? Or is it actually sometimes down even deeper? I just don't know.
Yeah. But
man,
that night of a foot. I love that so much. Yeah, that a lot left to be known, which is amazing. And shifting gears a little bit into just a final question from Lisa. Lisa says, mollusks are my jam, especially gastropods. I moved to the ocean about 2 years ago, and every single time I go to the beach, I find a species I've never seen before. Do you have any book recommendations for novice gastropod enthusiasts? And I'm going to add onto there any general tips for aspiring tide pool explorers out there.
For the books, Lisa, is Lisa from the West Coast? It's from depends on where you're from. So there's some really nice books for the Pacific coast of North America. Some general ones, some that are more books of keys, but have all the species illustrated. It really depends on where you live, Lisa.
Jeff said, or Lisa says that they're now living in California, San Pedro. So,
Okay. Yeah. Well, the Bible for West Coast tide pools is a great book called Between Pacific Tides. It has a lot of invertebrates, including mollusks. There are some picture books. There's the Light and Smith manual, which is a big thing with all the keys, but lots of illustrations. For the bivalves, there's Paul's monographs. Every one of those species is illustrated. There are those. There's quite a lot of books. And iNaturalist, if you take a picture of your find and post it on iNaturalist, it's getting really good at identifying the species and other people will find it and identify things for you. So there's that's one way of doing it. Also, tide pooling tips for mollusks. Different shores with different wave exposure will have different species. But personally, I go to the places that have fairly broad intertidal with some wave protection at low tide, maybe not at high tide. So you want the circulating, clean water. But when the tide's out, it's fairly protected area, and those are the really biodiverse areas. But you can find something in any habitat, on jetties, in muddy bays, on the sandy beaches. You just start exploring. It's what I've been doing since I was a kid.
Oh my gosh. Well, thank you so much for giving us that little bit of wisdom and for sharing about this amazing, amazing clam. We're so happy to have you on tonight.
All right. Well, thank you. It's my pleasure.
Yeah. And we'll go ahead and bring up our third presenter, Rachel. Hi, Rachel, you're on.
Oh, hello. You ready?
All ready. Take it away.
Hi. Hello. Greetings from Central Australia. My name's Rachel Poltridge. I'm a wildlife ecologist based in the desert. I've been here for about 30 years. And I currently work for the Indigenous Desert Alliance, which is a sort of like a support network for Aboriginal rangers. So I'm lucky enough to be able to work with these rangers on threatened species projects, and part of my funding comes from the National Environmental Science Program, specifically to set up partnerships between scientists and rangers to work on threatened species. Tonight I'll be sharing the story of the night parrot, Australia's most elusive bird. It's a short, fat, dumpy bird with a short tail that lives in this big spiky grass that we call spinifex. And it only comes out under the cover of darkness. So this species was lost to science for about 100 years, hiding away in the most remote corners of Australia. The elusiveness of the night parrot even features in the mythology of this species held by the traditional owners. In a creation story from deep in the desert, the Maru have tell this story of this being that wouldn't reveal itself despite fires, floods, hailstorms. Eventually it was banished to the darkness. Despite this, in Central Australia, up until about 100 years ago, Aboriginal people did used to be able to hunt them for food by tracking them to their spinifex tunnels that they were inside, using fire to flush them, and they did used to eat them. The first specimens of night parrots were collected in 1845, and it seems that they were relatively common in the 1800s. There was about 28 to 30 specimens collected in the sort of 1870s. And they were common enough in Alice Springs, my hometown here, that at the local telegraph station, there were picture frames on the walls that were adorned with night parrot wings, brought in by the station master's cat. But by the early 1900s, they were getting really hard to find, and the last specimen was collected in 1912. After that, there were many expeditions looking for them, but no further proof could be found, although anecdotal reports trickled in. Certainly there were no photos ever taken. In 1989, an entrepreneur in Australia called Dick Smith offered a $50,000 reward for anyone who could find evidence that night parrots still existed. And lo and behold, the next year, a dead one was found. And it was actually found by the curator of birds from the Australian Museum, who apparently stopped out in the middle of Queensland on a remote road and found a dead one on the side of the road. I bumped into this guy a few years later, and he was out spending his money on a night parrot expedition in Western Australia. And I was like a little bit skeptical, like, of all people, he'd be the person who might have access to a carcass. Did he just plant it to try and get the money to fund some research? But no, of course not. And about 10, 15 years later, another dead night parrot turned up in the same area, this time impaled on a barbed wire fence. So now we knew night parrots were still around. There was only dead ones found, but this obviously stimulated a lot of excitement, and a lot more searches were held looking for them. Nobody searched harder than this bloke, John Young, a bird expert, an old school egg collector, a highly skilled naturalist, and a host of a TV wildlife show. He went back to the spot where the dead one was found and searched, and apparently heard whistles, heard their calls, he thought, back as far ago as 2007, but it wasn't until 2013 that he finally actually found some living birds and got some photographs of them. John's a larger-than-life sort of character, a complex character, very charismatic, but quite controversial. After he'd made this amazing discovery, he couldn't help himself. He had to keep impressing people, and he came up with more and more claims of finding more and more birds. There were, so many of them out here. They were on all these different habitats and getting photographs of nests. But sadly, a lot of these claims turned out to be false, even to the extent that he was a nest that he'd taken a photo of apparently had fake eggs inside. So yeah, although he had made this great discovery, no one could take that away from him, that he had rediscovered the night parrot and found, the first evidence of living, breathing night parrots for the first time in 100 years. I heard about these photographs before they were made public from a colleague, Steve Murphy, pictured here, who had been sort of researching night parrot records for a long time. Steve's a meticulous ecologist, very highly regarded land manager and a bird expert. So he was the only scientist that John Young would trust to take him to the site and show him this population of night parrots that he'd found. So Steve and his wife Rachel became the first scientists to actually study night parrots. They studied their behaviour, they monitored nests, they did more surveys in the area. They even tracked some birds, putting little trackers on them to see where they went to feed, and they worked out, what sort of habitats they needed and what their threats were. I was invited onto the advisory team, which ended up becoming the recovery team for this species, due to my expertise in predator management and also my experience and networks with Indigenous rangers across the desert. Another thing that Steve and Rachel did is to secure this night parrot site in Queensland as a conservation reserve with the non-government organisation Bush Heritage Australia. Steve eventually took on a PhD student, Nick Lesberg, pictured here, and he continued the work for the last 5 years. He's been the one monitoring them, doing more surveys, studying the birds. And what Nick and Steve found was that night parrots obviously stayed in this big spiky spinifex grass in the day, but at night time they'd emerge just after sunset, and as they came out, they'd give a call. They'd give this sort of characteristic whistle. There's a few different calls, but it was quite reliable that if night parrots were in an area, they'd call just after sunset. So they were able to develop this survey technique based on this, using these little machines called song meters, acoustic recording units that you could set up, make them turn on for a couple of hours after sunset. And if there was a night parrot roost within a few hundred meters, you'd detect it on these song meters. So this became the tool we could use to roll out surveys across the rest of Australia to see if there were any more populations. But where to look? Australia is huge. The deserts are enormous, remote. The spinifex goes for thousands and thousands of miles. So where would you put these little detectors? Most of the scientists live around the coast. Most of the desert's got no roads. There's no good maps to work out what sort of vegetation's where. So it was a bit of a tricky thing. Where do we start? Fortunately, we've got this network of Indigenous ranger teams across the desert. 60 teams working out in these remote desert areas. Aboriginal people that grew up on country know their country really well. They can travel through the bush without roads, without maps. They know where different habitats are. They're funded by the government to protect their country, to protect the natural and the cultural values of their land. So, you know, they do things like fire management, weed control, threatened species surveys, feral animal control. Indigenous rangers slowly and quietly started finding night parrots. The first one was a blurry green image on a camera set by these Paraku rangers in the northern deserts of Western Australia. They'd set it at a water point that they knew about. They followed this up with setting out those recording devices and getting calls. And then they also set a camera at this little puddle that only lasted about a week. And they thought this might be where a night parrot will have a drink. And sure enough, got a photo. That was in 2017. Just before this, some bird watchers had actually found the first record of night parrots in Western Australia at another site and captured this beautiful photo when they flushed it of a night parrot flying through the spinifex. But it was once an Indigenous ranger group found night parrots and started talking about them at these big sort of ranger meetings that occur in the desert, that other rangers started taking notice and thinking, oh, maybe we might find them too, thinking about the kind of country that they had on their lands and where the right habitats might be. So it inspired a lot of other surveys to happen. And so now we've got about 7 different populations around Western Australia. But the biggest population has been found by these guys, the Ngurapa rangers in the northern deserts of Western Australia. And night parrots were first found on their country on one of these song meter recorders in 2018. Since then, they've continued to do more surveys every year. Sometimes using a helicopter to get to really remote areas to put out these little machines. And each year they've found more and more sites while still monitoring the original sites that are still persisting. They've now got 11 different sites across their IPA, Indigenous Protected Area, that span an area of 160 k's from north to south, 50 kilometres east to west, 11 different spots around there, including 5 roosting areas where you can reliably hear night parrots every night, and then other feeding areas. So this is the biggest known population in Australia. The Ngurapa Rangers, like many other Indigenous ranger teams, use a two-way science approach to their work. So this means combining that traditional Indigenous ecological knowledge with modern technology and scientific methods. And here we can see Clifford Sunfly, one of the younger Ngurapa Rangers, who has become a key researcher or key player in the night parrot research. Clifford grew up in his remote community watching David Attenborough documentaries, and so he's quite worldly in his knowledge of wildlife around the place, more so than most people in communities. And but he's like really likes getting into detailed work, and he'll search for scats and feathers and tunnels in the spinifex. You can also see here Dr. Angie Reed. She's actually from the US, came out to Australia to do her PhD, and then has taken on the job as the coordinator of the Ngurrapa Indigenous Protected Area. Angie's an excellent operator in the field. She runs a tight ship on the field trips, makes sure all the equipment's well maintained, deployed, properly. She's very rigorous in her work, and together between the rangers and Angie, they're doing a fantastic job. One aspect of the two-way science that we're doing is documenting information about the food and water resources for night parrots out in this country. And we're really lucky to have the chance to work with these ladies, Yati and Yukuji from Kiwikara country, who grew up in this land where some of the night parrots are from. They actually were part of the last family to grow up completely traditionally out in the bush, living off the land. So they grew up on these seeds. They've got an excellent knowledge of which seeds need fire for them to grow, which plants you don't want to burn, where you find the last seeds in the driest times, where the waters are, because they depended on it for their life. So we're assuming that a lot of the seeds that they used to eat in this country will be the same ones that night parrots eat. So it's a really good way of working out how to manage those resources. Of course, we don't know for sure exactly which seeds the night parrots eat. So what we also want to do is collect some scats from their roosts and get DNA analysis done to find out exactly what they are eating. But it's likely they'd be eating these same palatable seeds. The two main management issues for night parrots, they're threats, fire burning out those that habitat that they rely on, the old growth habitat. We have a lot of wildfires started by lightning, so the rangers do burning in the cool season to stop those big fires. And they do that by ground burning as well as aerial burning, dropping incendiaries from a helicopter. You can see here the rangers are planning with Angie their fire burning they're going to do that year. So they use satellite imagery to work out where the high fuel loads are, where it needs to be burnt, and they can do some quite fine-scale protective burns close to the night parrot habitat, and then sort of more broad-scale burning further out. The other main threat is feral cats, especially as young birds are starting to fledge. They'll come out of their spinifex in the evenings before they've learned to fly properly, and apparently they're very vocal at that time, which attracts the cats and makes them vulnerable to being eaten. I should have said that other predators of night parrots that they found at that Queensland population was a snake came and ate some of the eggs. Some of the fledglings from the nestlings died during a heat wave as well. So that Queensland population that's been studied for a number of years now is still very small. They only know of about 5 pairs of birds. It might only be 20 birds in that whole population, whereas there's probably going to be a lot more here in Western Australia. But back to the cats, feral cats are actually a favoured food source for Aboriginal people, some Aboriginal people out in the desert, especially these ladies that grew up in the bush, and they're highly skilled at tracking cats down and hunting them for food. So that's one method that is a successful control method. But out in these night parrot sites, they're a long way from where people are living, and it's a bit hard to do that traditional hunting regularly enough. So this year, the Ngurrapa Rangers are going to be trialling this special machine called a Felix, a grooming trap, that's been designed to be a very target-specific method of cat control. So what happens is the machine can recognise a cat from any other species. If a cat walks past, it'll shoot a jet of poison gel onto the cat's fur, and the cat will then compulsorily groom itself and lick off that poison and die that way quite quickly. So that's what we'll be trialling this year. So going forward, we'll be continuing this research. We've got this funding from National Environmental Science Program to build these partnerships, continue this work with the Queensland scientists, but maintaining it as a ranger-led research project, the way the rangers want to do it. But through that work, we're hoping to study the diets, feeding behaviour of night parrots. We're hoping to get Steve over to help us put some little trackers on the birds and find out where they go. We're going to try and make a habitat model that will help us search for night parrots in other areas. And then we'll just continue sharing this knowledge with other rangers, scientists and the government to ensure that Australia's most elusive bird does not disappear again. Thank you.
Thank you so much, Rachel, for sharing this incredible story and the work you're doing with the Indigenous Desert Alliance. These partnerships and this work is all really inspiring. I'd like to encourage everyone in our audience to continue to ask the great and thoughtful questions that you've been asking all night for Rachel, and we'll get to as many as we can. Firstly, knowing the vastness and the remoteness of the spinifex habitats that you mentioned in the Australian desert, how likely do you think it is that there are further night parrot populations that we aren't yet aware of?
Yeah, I'm pretty sure there will be quite a few more populations turn up. I mean, we've only surveyed a very small portion of the habitat so far, but there are a lot of ranger groups going out and trying to pick the best habitats and finding them in some areas and not others. But the more we can refine our sort of habitat model and know exactly what kind of spinifex to look in, the better. But yeah, there'll definitely be more populations out there in Western Australia. It seems like that there is a lot of good habitat for them. Yeah, and the more sort of fire management that's being done by the rangers to make sure there's always old growth spinifex left, the more chance that night parrots will be able to persist out there.
Great. James wants to know if there's any kind of estimate for the population of feral house cats as like a major threat. Is there any understanding of like how much progress is being made?
So the number of cats in Australia, there has been an estimate on that, and it's something like about 5 million cats in Australia, 4 or 5 million. So they're everywhere. They're in every habitat of Australia, every corner of the country, from the driest deserts. They don't need water to drink. They can just live on the blood of their prey, I suppose. And they threaten a lot of species. So they're a major threat to wildlife in Australia. There's a lot of different control programs going on for them, but you know, it's such a huge country, it's pretty hard to do anything that's very broad scale. But in some areas there's baiting, in some areas there's trapping, shooting. There's a lot of effort goes into cat control, and we're still fighting a losing battle in a lot of places. But in these little discrete areas where you've got a ranger group on the ground that can keep up the control ongoing, it is making a difference. And a lot of our threatened species are surviving in these little pockets where they've disappeared in the wider surrounding areas.
Yeah, it does sound like an intimidating undertaking, but it's heartening to hear that under the right conditions, the control efforts are effective. A couple people in our audience are asking if the method of cat control that you mentioned, where the poison gets shot onto the fur and then the cat grooms itself, is there any worry about introducing that poison into the environment, say like if a predator were to eat that cat? Or
not really. That poison breaks down very quickly in the cat's body. It gets metabolized, and so there's not much risk of secondary poisoning. There's not many animals out there that are going to eat a dead cat anyway, mainly maybe a dingo. But it's compared to everything else, it's very target specific rather than, dropping baits everywhere or, so it's the most target specific method we can have. And
Oh, great. Outside of feral threats to night parrots, are there any known native predators?
So I mentioned the snake that ate the eggs is one predator. What else would there be? Possibly some birds of prey. Most of them are diurnal rather than nocturnal. I'm not sure if an owl would get a night parrot or not, but some of the birds of prey do hunt around sunset. Possibly goannas. Some of the big lizards might come into their nests and get the eggs. But I think probably cats and foxes are their main predators now.
I know you mentioned a couple of times that the night parrots have tunnels. Are these something that they dig themselves? Do they use them for nesting or just for shelter?
Yeah, yep. So they make them themselves. They snip off the spinifex going into the tunnel. It can be, you know, quite a way into these big spinifex clumps. So they have both roosting tunnels and nesting tunnels. The roosting tunnels are like closer to the ground, whereas the nesting tunnels might come in from the top of the spinifex and be sort of right in the middle of that spinifex. But yeah, they make them themselves, and that grass is very spiky, so it's very good protection for them.
Oh, so they tunnel into the vegetation itself, not like underground?
Yeah, no, not underground, into the spinifex grass itself. Yeah.
Interesting. Blair asks, what other parrot species are the night parrots genetically like closely related to?
There's another species called the ground parrot that they're very closely related to. So they occur down along the coast in Australia, and yeah, they are similarly threatened by the same things, predators and fire, I think. So they're quite rare as well, but yeah, they are quite similar to the western ground parrots. And I think there's eastern ground parrots as well.
And are similar conservation strategies being employed for the ground parrots?
Yeah, I know they're definitely doing cat control down around parrot habitat. And yeah, I think fire management as well.
So the night parrots, do they move within their habitat very often? Do they stay pretty
Yeah, so we don't really know about that. That's what people assume, that they would be quite nomadic and follow the food resources as it shifts around. But the population in Queensland has been very sedentary, and I guess people have been surprised that they haven't found them in other sort of places that they've moved to. They seem to just be in this same little spot, and it might be the same in Western Australia as well. That certainly for the last few years, the ones we've been monitoring have been at those same spots year to year, whether it's a good year or a bad year. So perhaps they're not as nomadic as other parrots, like budgerigars, for example, are very nomadic across the desert. But it seems the night parrots do have these, favoured areas where there's probably good feeding grounds nearby, there's good shelter, and there's water. If they can find a spot that's got all those things that they need, they possibly can just stay there. But, we don't know enough. We need to monitor these species for a lot longer to really know that for sure.
Yeah, definitely. And then one last question before we wrap up for tonight. Through having these Indigenous ranger groups on the ground, have we learned anything more about night parrot behavior that can help to influence their conservation?
Yeah, it's a good question. I mean, there doesn't seem to be a lot of knowledge about their behavior from the rangers, because they have been so rare for such a long time. But definitely there's that knowledge of the food resources, the water resources, how to manage the habitats, that sort of thing that we're really learning from the rangers. So I guess we haven't learned so much about the behaviour yet. It's more a case of rangers themselves learning it now as they study them themselves, rather than knowledge that's been passed down. But yeah, definitely we're learning a lot about the other animals and plants that are around that are helping the night parrot survive.
Excellent. Thank you so much for sharing.
No worries. Thanks for having me.
Of course. Okay, so in closing, we would love to give such a massive, massive thank you to Stephen, Jeff, and Rachel. To hear your stories was phenomenal. I'm so happy to be back. This is a great reintroduction to night school.
Absolutely. And starting in March, we will be back the third Thursday of every month. Yes, that's the third Thursday now. Things change. Bringing you all of natural science's coolest researchers, creators, and innovators. Our next program will be full of crap. And by that, I very specifically mean scat and feces and waste and poop and all of the things that we can learn from it. So please join us for our second Night School of 2023 on March 16th for a program that'll make you and us very, very grateful that smell-o-vision is not real, which we're calling Night School number 2.
It's funny. As always, subscribe to the California Academy of Sciences on YouTube so that you'll be notified every time that Night School is in session. Recordings of every Night School stay on our YouTube in perpetuity, which means they never go away. So you can feel free to rewatch old episodes at any time from anywhere on Earth. You can send them to friends and family. You could like stream them at a library. I don't know. It's like spread the word.
The world of your teeny tiny clam, you know.
Make that clam famous.
But yeah, thank you all so much for joining us again. Take care. Have a good night.
Good night.